All right, good morning, everyone. So it's 8 o'clock in the morning. So welcome, everyone. Thank you for joining us again in our regular PAASE webinar. So I hope that those of you from the Philippines are now recovered from the typhoon last week. So today's lecture is about induced pluripotent stem cells, disease and tissue modeling, gene editing, cell therapy, toxicology, and drug candidate screening. Our speaker for today is a staff scientist at the National Institute of Mental Health, National Institutes of Health in Bethesda, Maryland, United States of America. She received her bachelor's in chemistry at the Mapua Institute of Technology and her master's and PhD degrees in biochemistry at the George Washington University and Howard University, Washington, D.C., respectively. She pursued her postdoctoral fellowship at the Laboratory of Biochemistry, National Cancer Institute, NIH, and subsequently moved to the NIMH Intramural Research Program. She has mentored postdoctoral fellows and students, three of whom are graduate students from the University of the Philippines. She conducts research with the goal of understanding the genetic underpinnings of mental illness that includes the central objective of identifying perturbed genes and molecular pathways that are crucial to disease etiology. Mental disorders comprise one of top causes of disability worldwide, requiring effective management and medications. To fully define mental disorder dysfunctions in human brain biology caused by genetic defects remain to be clarified. The inaccessibility of living human brain cells creates strategic challenges that when warrants studies on model systems. To create a cellular model, Dr. Seville employs induced pluripotent stem cells that are generated by reprogramming somatic cells from patients and individuals in the study. So I'd like to welcome everyone. Finally, we're pushing through with this lecture. So let's all welcome Dr. Sevilla Detera-Wadleigh.
Thank you very much, Kay. And thank you all for having the patience. This has been postponed twice, but okay. And I'm happy to be here and finally get this lecture going. A disclaimer, I have nothing to declare. What's going on? Okay. I thought that I'd start this talk by paying tribute to a number of people. In the spirit of the month of November, when we Filipinos usually devote special acts of remembrance for our loved ones who have moved on to immortality, I'd like to dedicate this talk to the following. Those who have passed away due to natural disasters, and there's quite a number in the Philippines, and COVID-19, my mother, father, sister Rosarina, and brother. And our very dear friend, Julie Ramos Asfour of Fort Erie, Canada, who passed on shockingly on Monday, November 16, 2020. We had no expectation that she was ill, and therefore it was a shocker for all of us. She was a PAEC co-worker from decades ago. She had BS in chemistry at Adamson University, MS chemistry at the University of California in Riverside, where she met her husband who was doing his PhD in soil engineering. She was an exceptional wife. Oh, wow. I'm sorry. Where are we? Sorry. Can I return?
Yes, just move.
And mom, and in fact, her eldest son has a PhD in chemistry, and who's my godson. She's very energetic, a magnificent teacher, a gourmet cook, a superb and wonderful friend with a natural ability for growing plants. And the husband told me that even on her last day, she was talking about the garlic in her backyard. a first-class mind with an insatiable thirst for knowledge. Julie, we miss you very much. So this is the outline of my talk. First, I will introduce the talk by presenting the global burden of disease. My research for decades has been on diseases, especially mental illness. And then I will introduce iPSC, Induced Pluripotent Stem Cells. This is not the stem cells that is popular among some actresses or actors and stuff like that. This is derived from mature cells. I will present some experiments on disease modeling using neurons, 3D brain organoids, cardiomyocytes, and also present one very recent publication on COVID-19 using iPS. Also present a study using CRISPR-Cas9 and some clinical trials using iPS for cell therapy. And then I'll have a take-home message. Okay. Here's the global burden of disease pre-COVID in 2017. This is measured using disability adjusted life years, which is the sum of years lost due to premature death and years lived with disability. So one daily equals 1 year lost of healthy life. And as you can see over here, when you have a light yellow, there's like 10,000 or less daily per 100,000 individuals. And Australia, you look at Australia, the Western Europe, and also Canada. You look at the Philippines, we are in about over 30,000 dailies per 100,000 individuals. The difference, of course, is the healthcare system. So what causes all these dailies? In 2017, the major cause are cardiovascular diseases, followed by cancers, neonatal disorders. This is red because it includes communicable diseases. And look at mental and substance abuse disorder. It's like the fifth cause of daily worldwide. And that's where my research is focused on. Now that we have COVID, the pandemic, I thought I'd present this graph also because it includes the Philippines. And this was done very recently, 12th of November. And you can see, this is from, you can see that the Philippines has about less than 100 people, less than 100 deaths on November 12th, 2020. So that's not terrible, but compared to South Korea, of course, there's a big difference. But the United States is still on the top. It's over 1,000 deaths on November 12. And the projection by Dr. Murray at the Institute for Health Metrics and Evaluation at the University of Washington is that by March 2021, if the mitigation efforts are not followed, we will have over 2.5 million deaths worldwide. So now let me, after talking about this global view of disease, I'd like to talk about human genetic diseases. And there are two types. One is a single gene or Mendelian disorder is caused by mutation in a major effect gene. And an example of this is well-known sickle cell anemia, where which is autosomal recessive disorder, and the mutation is on the beta globin gene. And most of the common diseases are complex and they are polygenic, meaning they're caused by multiple genetic variants, with each of these variants conferring only a small contribution to risk. So it's accumulation of these variants that cause diabetes, cancer, asthma, and neurodevelopmental, neurodegenerative, neuropsychiatric disorders. So how do we understand disease? I have put together a some sort of a flow chart over here, and it's you need to have human samples, a collection of affected, either cases, unrelated cases, or families with affected individuals and unaffected individuals. And this is, you do association analysis or genome-wide association, and you find the genetic variants that cause. It's usually when you have a complex common disease, you need tens of thousands of individuals in order to have statistically significant results. And you can do patient stratification once you have the gene variants or risk prediction. But then you need models here in order to understand what those gene variants do. You need models, and you do multi-omics analysis, you do phenotype assays, and then hopefully you get the you identify or you define the perturbed biological mechanisms. And from there, you probably can do diagnostic and prognostic markers, therapeutic targets, and what you can start thinking about what environmental risk factors are there and maybe preventive measures. So going back to the global burden of disease again, understanding disease could lead to a reduction in disease burden. When we understand diseases that could lead to a reduction in disease burden, improve people's well-being, and create advances in the economy through disease prevention, effective management, and successful intervention. I'm sorry. Oh, I don't know why this is. So I was talking about disease models. You need disease models because, if you want to study brain disorders, you cannot take living brain cells from living human beings, and you cannot take living heart cells from humans. So you need to, it's inaccessible, in other words. So, but then, because they are models, they don't really fully recapitulate the hallmarks of the disease, but they are important in order to understand the biology of the disease. And so the usual animal models are flies and mice, zebrafish, and cellular models. For a long time, people have used immortalized cells. But since the 2007, the discovery or the development of iPS we can have living human cells which are human derived and patient specific. And the scientist, okay, so I'll show you later on what is iPSC. They are pluripotent embryonic-like cells generated by reprogramming somatic cells from mature tissues. I'll show you the next slide. And this was done by Yamanaka and Takahashi, first in mice in 2006, and then in human cells in 2007. They used fibroblasts to convert them to iPSCs using 4 transcription factors. These 4 transcription factors are proteins that regulate the activity of other genes. And for that work, Dr. Yamanaka got the Nobel Prize in Medicine or Physiology in 2012. So iPSC is truly a game changer. What you do is you start with mature cells like a skin or blood cells, and people have even used urine and generate iPSCs. The other advantage of iPSC is that you can take that skin cells and convert it to iPSC, and so that carries a copy of that donor's genome. And so it's either patient-specific or donor-specific cells. So when you have the cells, you're studying the cells of that particular individual. And because iPSCs are pluripotent, they can be differentiated to disease-relevant cells, like which are inaccessible in living persons, as I said earlier. So they can be differentiated to brain cells, heart cells, liver cells, etc. And they can be expanded, stored, and recovered, so you have a continuous supply of cellular reagents. So what are the applications? As I mentioned, cellular platform for disease modeling, for genome editing, tissue modeling, drug effect and toxicology screening, screening for candidate therapeutics, and cell therapy. The advantage, as I said, is because it's taken from that same individual, it's autologous, so there's no immune rejection. So how do you generate iPSC? I have a diagram here. So you can take a skin biopsy from a donor, a patient, and we take that to the lab and convert that to fibroblasts right here, these elongated cells. They get converted after about a month. So you're gonna see cells that look like this, and you can fix them and hybridize them with antibodies, which is then hybridized with a secondary antibody that carries a dye. That's why you have green, red, and the blue is the DAPI that hybridizes or binds to the nuclei. So then you reprogram, but as I mentioned, you need these 4 transcription factors. And after about a couple of months, you see that these elongated cells get converted to colonies that look like this. Each colony contains individual cells, as you can see over here. So as I mentioned, you can also start with blood cells, PBMCs, and you will get the same thing. And these are done in culture dishes like this. So that's why we say that these are your cells being grown in a dish. The other thing that you do is you make sure that the iPSCs show normal karyotypes. If they have abnormal karyotypes, then you don't use them for further studies. So I'm not gonna talk about some examples of projects that are being done to study the workings of the human brain. And this is normal brain. You're familiar with the different lobes of the brain. And what is interesting is that the adult resting brain uses up to about 20% of the total body energy. So we need to take care of our brains. Diseases involving the human brain. Examples, this is not a comprehensive list, of course. I'm just listing 3 different major diseases: neurodevelopmental, autism, or Smith-Magenis syndrome. This Francis Gordovis in the lab studying this SMS. Neuropsychiatric, I've been studying bipolar, and schizophrenia have now been found to really share a lot of genes. Neurodegenerative, examples are Alzheimer's and Parkinson's. And here are examples of a healthy brain, and after severe Alzheimer's, you can see the shrinkage. And what are the major brain cells? Neurons, they are the basic working units of the brain. And there are about 86 billion of them. The synapse, that's where the neuron connections occur, where signals are both received and sent out. And there are trillions of synapses. You have the glia, which are the supporters and protectors of the neurons. Astrocytes are star-shaped. You have microglia, which directs immune response, and oligodendrocytes. They insulate the neurons, the axons, and they form the myelin sheath. So here's just a cartoon of a neuron with the synapse right here, some neurochemicals. The vesicles are putting out the neurotransmitters. And this is the postsynaptic cell. So that's how our neurons communicate with each other, and they communicate with muscles and other cells. So when you differentiate iPSCs, these are the colonies right here. Provide the iPSCs with nutrients and other chemicals in the cocktail. And one of the earlier cells that is produced are these neural rosettes. They're called neural rosettes because they look like roses. And then you get these neuroprogenitor cells, and then the neurons. See, here's the cell body, and those are the neurites. And the first time I've produced myself neurons in the lab, I was really, it really amazed me, because I said, if the connections of the neurons are like this in a dish, how much more it is in our own brain. So this is, again, an example of the differentiation from iPS. You get neuroprogenitor cells and then neurons. This is in schizophrenics, and this is in controls. There was an assay that these authors did, and there was a difference in the connection between the control and the schizophrenia derived neurons. So in order to study the disease, this is a cartoon of what types of assays and what types of methods do you use in order to model the neurodevelopmental, neuropsychiatric, or neurodegenerative disorders. So as I said, this is iPS. You produce different types of neurons and other brain cells. And then you do these assays, RNA-seq, where you are looking at the, analyzing the transcriptome, or what the genes are producing. Genes transcribe, DNA transcribe to RNA, RNA translate to protein. So this is RNA-seq. You can capture the transcripts from these cells. You can do bulk RNA-seq. Now, single cell RNA-seq has got very popular. You can do 3D epigenomics, where you can assay for DNA methylation, and multi-electrode array. What you're doing there is determining the action potential, the activity of the neurons, also with electrophysiology. This is using patch clamp, that's for individual neurons, and of course microscopy in order to assay the morphology of the neurons. And these are the other things that you can do. So we do this in the lab. And we're starting with the MEA now. We have a new instrument, and hopefully we can get the things to work. So as I said, most of these are done on a monolayer culture, but it's having three-dimensional brain organoids, studying them, generating them, is getting more and more popular, because with brain organoids, you can mimic the structure of the brain. With monolayer cultures, you only have, excuse me, neurons and astrocytes in a dish. But with 3D brain organoids, you can have different structures. You can have the hippocampus, you can have hypothalamus, etc. So I'll introduce 3D brain organoids. This is a good cartoon showing human brain development from day 1 to 39 weeks. This is what you see. This is the cortical plate. This is and then you have the brain organoids. It takes 17 weeks to get a cerebral organoid. And as you can see, this thing seems to recapitulate what you see in the human brain. Ventricular zone, subventricular zone. But now people are growing cerebral organoids up to hundreds of days. So this is the first brain organoid that was published by Lancaster et al. In 2013. And it does mimic human brain development as shown here. And Kay asked me, what is the size of these brain organoids? They're about 4 mm in size, millimeters in size. And the actual size is right here. And this is a cartoon of the human brain. This one sort of recapitulates what the human brain has, but it's still not the whole. The structures are not the structures of the human brain are not fully recapitulated here. But people are getting more and more sophisticated in generating brain organoids. And after you have brain organoids, you slice them up and then stain them with these different antibodies. Here's the forebrain, the choroid plexus, and these are the other markers that they use. This is for a more mature neurons after 75 days. And this was the cover of Journal of Neuroscience early this year. This is again after slicing the brain organoids, and it's immunostained with these different antibodies after 28 days. One of the first things that was studied with brain organoids is microcephaly. Here you have a patient with microcephaly, as you can see, small brain, and the control is a large brain. And the gene that is mutated is the CDKRAP2. As you can see over here, the protein is absent in the Western. And you can see the difference. Here's the organoids for the controls at different days. And this is for the organoids generated from the microcephaly. So it's directly from the patient and also from the control. You can see here the control versus the organoids from microcephaly after immunostaining. And microcephaly was also studied in mice, but they couldn't really see the major defect, but brain organoids was able to reveal this defect. So this is just a cartoon of all the applications that a human organoid can be used for screening for genetic variants, metabolomic analysis, drug development, cell therapy, genetic engineering, etc. Now I'd like to show you examples of the use of our iPS in cardiovascular research, because here's the heart, of course, the normal heart, the ventricles, and the atria. And with iPS, you can generate all these atria and ventricular types of cells, atrial cells, and nodal cells. After 60 days, they look like this. After 10 days, I'm not a cardiovascular researcher, so, but you can see the difference when they get more mature. And after a certain period of maturity, you can see that in the dish, the cardiomyocytes beat. So you can see here. And with labeled cardiomyocyte, it's you can even see it better. Now I'd like to switch to talking about genome editing. There's there's quite a bit to cover. You must have, known about Jennifer Doudna and Emmanuel Charpentier, who both got the Nobel Prize in Chemistry for CRISPR-Cas gene editing. And apparently some of our PAASE members have collaborated with Jennifer Doudna. And of course, Ben DeLumen knows her because she's at Berkeley. So this is this is CRISPR-Cas editing. What happens is that it is very powerful and it is also easy to do. If you want to make a cut in any part of the genome, you can do it by having a guide RNA that matches that part of the genome. And in the presence of Cas9, that's the one that cuts the genome right here, double-stranded break, and it's then repaired over here using HDR. That usually they don't use this because there's more random mutations that occur. So we have used this to correct a mutation in SMS. And Francis is studying that. It's an ongoing project. So it is so powerful that Jennifer Doudna had to convene a conference of people in the field, because this can be used for, if you can edit any place in the genome, you can think of eugenics. You can think of all kinds of things that people can do. So there has to be a control in the use of this new technology. I want to present just an example of where CRISPR-Cas was used. And there's so many publications now using CRISPR-Cas. But there is a pathogenic copy number variance on 16p11.2. This is present in autism and schizophrenia, in bipolar disorder, and intellectual disability. So this is 16p11.2, and some autistic patients have deletions here, or some bipolar patients have duplications here, and it involves 29 genes. So the question is, what are the neurobiologic function of these genes on 16p11.2? So these authors presented this new paper is by Kistner et al. I forgot to put it in here, where they used CRISPR-Cas to knock out one of the genes, which is KCT-D19, and knock it out in iPS-derived neural cells, in iPS, and then you differentiate into neural cells. As you can see here, this is the control, and these are the neuroprogenitor cells. You can see the proliferation. When it is heterozygous, meaning one of the copies is knocked out, there's fewer cells. And when both copies are knocked out, there's also fewer cells. And you can see also when both copies are knocked out, there are fewer neurites in the neurons. So with CRISPR-Cas, you can show that you can sort of define the role of KCT D13 gene that is located on 16p11.2, and this is probably how this particular gene contributes to the pathology of, for example, autism or schizophrenia. Now, I'd like to I don't have enough time to show you all these experiments, but IPS has been used in COVID-19 cellular modeling. I'm just going to show you the recent paper using iPS-derived brain organoids. You are very familiar with this. COVID-19 virus and the receptors are ACE2 and also helped by this TMPR SS2. Those are the, if the cells have those receptors, then COVID-19 can successfully infect those cells. I'm not showing the lung or the heart, but I'm going to show you the brain experiment. And so what they did, Lancaster and Knoblich, and also is that they infected the cerebral organoids with the live virus. Oh, this is by Pellegrini, the drawings here are very Lancaster and Knoblich. Pellegrini et al. Infected it, one by infection with a pseudovirus, the other one with a live virus. And this is what they found. Here's the cartoon. The virus infected the ACE2 expressing epithelial cells in the choroid plexus. The choroid plexus, the part of the brain that produces CSF, cerebrospinal fluid, and cerebrospinal fluid protects our brain. And here is a cartoon where the epithelial cells, after infection with the live virus, they die. But what they did not see much infection with of the neurons and glia. So this is really an interesting finding. There was another group that also used live virus to infect brain organoids, and they did not see much infection in neurons and glia, but they saw, they found high amount of infection in the choroid plexus. So this is one paper that shows the use of iPS-derived brain organoids in COVID research. I'm now going to switch to examples of iPS-based cell therapy, and this was taken from a review by Yamanaka this year, and apparently there are clinical trials now in Parkinson's disease, macular degeneration, and all these other disorders using iPSC-based cells. And in age-related macular degeneration, which is the major cause of vision loss in people over 65 years old, there's the dry and the wet type, and there's no effective treatment for AMD. So here's what happens when the person suffers from AMD. Here's the normal photoreceptors, retinal pigment epithelial cells, and these other structures. And as shown here, the person with a normal vision can see this tree. But when there's cell death, the RPEs are dying. That's what you see, a black thing. You don't see the tree. So there's that vision loss. At NIH, Kapil Bharti has started a clinical trial for stem cell therapy for age-related macular degeneration, and he will enroll 20 participants starting in September, and the study completion will be 9 years from now, because what they are, the patients that they are studying for this particular cell therapy are those who have advanced AMDs. So hopefully that will be successful. And another sort of series of clinical trials is being done by the University of Göttingen. They will start in December, and 4 years from now. But what they will do is they will generate human myocardium from iPS, and then use that to transplant that onto the heart of people with terminal heart failure. They will enroll 53 participants, and hoping that that will work, because as you can see, the cardiomyocytes can beat spontaneously. And this is another clinical trial that is being sponsored by a private company, 5 participants, and they're just going to directly inject cardiomyocytes into the myocardium. So we will see what happens to that. I don't know if they've started, but they say the starting date was May 2019 and ending a month from now. So we will know what happens to this clinical trial. So the take-home message is that human diseases can be investigated using cells like iPS that are derived directly from the patients that you're studying. And that is the huge advantage. An iPS platform can be used to define disease mechanisms, screen for new therapeutic compounds, and the ones that I've already presented. The future use in autologous cell therapy, where there's no rejection or maybe minimal rejection, but I did not talk about limitations, but they exist. So before I end, it's very important that I acknowledge some of the people who I have worked with, and this is Philippine focused, not the people that I work with over here. UP professors Virginia Monhe and Amir Fina Santos, and my former PhD students, they were in the lab. Gloria Reyes, she was working at the Wellcome Trust in the UK. Delberto Ferraran, who is now a professor, has been a professor at the Visayas State University, and is one of the directors there of one of the centers. UP Manila professor Jose Nevado, who is the advisor for our current MD PhD student, Francis James Gordoves. He will be finishing at the end of the year, hopefully. He will have his PhD defense the early part of December. So I'm sure he will do very well. My PAASE colleagues who for decades I have been received their unshakable friendship and support. Funding sources for for students, the OST. My passive friends, I'm not going to mention any names because if I mention one name, I will forget the others. So my passive friends, they know who they are. And my friends at UP and Manila and UP Diliman. So I have included some pictures over here with Chancellor Carmencita David Padilla. She's the Chancellor of UP Manila College of Medicine. She was here. She told us what she's doing, all her activities at UP. And we were just so excited to learn about them. Here is Professor and Mrs. Flo Kyocho when I visited in Houston last year. By the way, Professor Kyocho and I share the same mentor. His mentor for his master's and my mentor for PhD, the late Dr. Felix Friedberg. And he always used to tell me about Professor Flo Kyocho. Look how successful he is. He worked with 2 Nobel Prize winners. He's doing all this X-ray crystallography and publishing in the best journals. I said, okay, well, he's a role model. So thank you, Flo. And so Francis, we were invited to give a talk in Houston at the Baylor College of Medicine last year at the SMS Research Symposium. And here's Francis, there she is. And these are a couple from the University of California in San Diego. And Anne Smith, she's very famous. She is the co-discoverer of Smith-Magenis syndrome, and she is our very nice collaborator. And here are some pictures that I found. There's Rosie and there's Giselle. Of course, my friend Lulu. This is one of the, I don't remember what year this was when they visited and we had a party at home. And there's the, what's that? There's the pig. This was in Tucson, Arizona, after the meeting. So here are some passive friends. And there's, I found Kay, she's there. And then Toby Dyrite came here and presented his work on the coconut. And there's Joey. There's the late, I forgot what was his name again. Say that again.
Blanche.
Yeah, Blanche. Catalino Blanche. She's a very funny guy. There's Lisa and there's Leah and Miss NIST.
Baltz Aguda.
No, he is not here. Okay. Well, thank you all. If you have any questions.
Yeah, we actually have a lot of questions. Thank you so much for that, Seville. You really, there's really interest in the group. Okay, so let me begin with a question from Rosalia. She's asking, what are the major artificial components necessary for the brain organoids to successfully propagate?
Oh, there are these, what do you call this? the problem is some of the cocktails, um, the components of the cocktails are not divulged by the companies, but the cocktails that we use for generating embryo bodies, and then from those embryo bodies, you have the spinner, and that's where the organoids are produced. I don't know the components. I don't know what they are. So
Okay, Rosalia, did you want to make a follow-up? Yes, so kindly unmute yourself.
yeah, turn on your
Yeah.
audio.
I was just wondering, are there any extracellular matrix, important in matrix that are, special to brain organoids as opposed to other like cardiac organoids or
Yeah,
organ?
actually they're floating, but when we grow monolayers, we have to use matrigel, but they use matrigel also in brain organoids. I don't know. So we use matrigel. You can use Geltrex, and for neurons, polyornithine laminin. Both the plates with them. That way, the things stick attached to the bottoms of the well. I don't know. I'm not sure.
Okay,
Thank
thank
you.
you.
Yeah, so Eva is raising her hand. I just want to ask Eva, is this a related question?
Say that again.
No,
Eva,
no,
Miss Eva, yeah. Yeah.
no, it's not a related question. It's another
Oh,
question. So I'll
Eva.
wait. Yeah. Hi,
This is
ma'am.
another friend from UP Manila.
Okay.
Yes, ma'am.
Yes, please kindly ask your question.
Okay. So first, let me congratulate Dr. Seville on behalf of Chancellor Padilla and the National Institutes of Health, UP Manila. Thank you for your excellent lecture and for also presenting to us the various applications of induced pluripotent stem cells. We look forward to Dr. Francis Gordovis coming home and setting up a lab like yours at the new NIH building, which is set to open late next year to early 2022. We hope the collaborations can continue. But on another on another note, Dr.
Right.
Steele, yeah, I'd like to ask, what are your thoughts about using genome editing in a uniquely Filipino disease called X-linked dystonia Parkinsonism?
How many? I'm not sure about the cause. What is the cause? Do you
The
know?
cause of the condition is it's a TAF1 gene mutation wherein there is a insertion, a retrotransposon insertion in an intron in the intron 32 of TAF1 gene, and so
How large is it?
it's about 2.2672 base pairs long, Dr. Seville.
Oh, okay. Well, what if you want to use IPS? This is it. If you want to use IPS to generate those types of neurons, so what you can do is you can cut the thing out, that transposon out, using
Mm-hmm.
CRISPR-Cas, and then with the with the transposon out, then it gets like the normal thing gets ligated together,
Mm-hmm.
and then see what happens. Whether you can, because what you want to do is to compare it to a cell that carries a normal that normal gene,
Mm-hmm.
and the cell that carries the abnormal gene. Now, if you cut that out, see what happens. Does it rescue with the, if you remove that transposon, does it simulate or recapitulate the normal morphology or whatever assay you can do?
Thank you. Thank you,
Okay.
Dr. Seville.
Okay, the next question comes from Vince Darius. It's quite a long one. Uh,
I hope I can answer it.
Yeah, so thank you for the nice talk. So he has a first question. You mentioned that you are now doing some MEA measurements with
Yeah.
neuron differentiated iPSCs. Have you compared the responses with neurons in acute brain slices, perhaps of rodents? How does an action potential look like? Does it have good expression of ion channels, sodium, calcium, etc., for it to function?
Oh, okay. We got our MEA, our multi-electrode array, only last couple of months ago. And we just set it up and we're just starting to use it. And in fact, we haven't seen action potential yet. So we are at the very beginning stage. Um, we have collaborated a few years ago with people upstairs at the NINDS, and they did patch clamp on our iPS-derived neurons, and we saw action potential. But that was it. And the reason why we purchased this MEA multi-electrode array is that we want to see how our neurons what do you call this, how they behave. And the other thing that we are very interested in doing is treating the neurons with different kinds of drugs. For example, neuroleptics. What happens to these neurons? And I was listening to a seminar by Sergey Pasca, who's at Stanford Stem Cell Center, and he says that action potential, or MEA, is one of the most stable phenotypes in iPS-derived neurons. So we're looking forward to getting our neurons and our equipment going more, like as well as we could expect.
Okay, he actually has a second question. Rather than using 3D organoids, we have simplified the problem and cultured neuronal networks on nanostructured surfaces. So this is in 2D that mimic the structure of the extracellular matrix in the brain. So he also attached a link to a publication. We have performed calcium imaging and our neuronal networks function well, good synaptic transmission between cells. We hypothesize that the biomechanical cues that the cells experience while growing on nanostructured surfaces can aid in the growth of neurites similar to that of the brain. We also hypothesize that geometry of the nanostructures can define the type of neuron. Would it be interesting to see if iPSCs differentiated to neurons more efficiently on our surfaces?
I'm not familiar with your system, so I cannot make an intelligent comment. Did you publish this yet?
Vince, would you like to unmute yourself?
Did you publish your
Yes,
system
yes.
yet?
There's a link to our publication, and the neurons are actually having good morphology in terms of neurite growth.
How did you generate the neurons from iPS?
No, we it's a culture from rodent, from hippocampal neurons.
Oh,
They're
okay.
primary cultures.
Okay.
Yeah, maybe you can discuss further.
Right, that's right. That would be very interesting. So,
So yeah,
what are
we
you
were.
studying? What are you studying?
So we the main aim of that project is to actually understand how we could provide we could make neural networks networks of neurons that are more functional. So we hypothesized that if we grow them on a certain set of surfaces, not just a flat surface. I assume you're growing them on a cover slip or on a plate that's
Okay,
just flat.
right.
We thought that the neurons in the brain actually grow on a more has a biophysical property that they could actually be guided in terms of growth. So we thought that if we were to simplify the problem and grow neurons on nanowires rather than a flat surface, we can define the spacing of the nanowires. We can define their structure, and we could actually program their growth in terms of how they look like. We can, yeah, that's our.
Can I? When did you publish, or where did you publish it?
We publish it in Nanoletters. There's a link.
Nanoletters. Okay. Okay.
Yeah, I can get you in touch. Well, you're both in the PAASE Gmail, yeah, but I can link you up.
Yeah.
All right.
So I think you'll have a copy of this of the text. I think I sent
yes,
also
yes.
a link to.
Oh, okay. You can
paper.
send it out. Thank you. Thank you very much. Where are you located?
In Australia.
Where in Australia?
In Canberra, Australia National University.
Oh, okay.
Okay, I'll take a question from the YouTube. So this comes from Angeline Lau. So in Yamanaka's 2020 report, the Alzheimer's disease has not been included. What is the current status of applying iPSC for Alzheimer's?
Well, there's a lot of studies on iPSC-based studies in Alzheimer's, but not being in that field, I cannot tell you the studies that are being done. But what I did not include here was one study in Alzheimer's where they generated different kinds of neurons, and they were looking at the endosomes in the neurons, and apparently there was a difference, but I did not read it very carefully. So.
Okay, thank you for that, Seville. The next question comes from Anna Serkina. So there have been reports of long-term post-COVID symptoms, including brain fog. Do you have any hypotheses regarding this? You showed how SARS-CoV-2 destroys epithelial cells in the brain, but not the brain cells themselves. She also has a follow-up. Also, umbilical cord banking is a very expensive procedure. Do you think that iPSC can replace umbilical cord biobanking?
Oh, I have no idea. The second question, I have no idea, but people are generating iPSCs from cord blood. So that's that much I know. As far as, this is the reason why people were interested in doing research or investigating brain organoids that have been infected with COVID. It's because some people have seizures, they have headache, and as Krista says, they have brain fog. So we don't know what causes all those brain fog, but that's one of the things that they found. And this is actually a paper by Lancaster's group. Lancaster is the first person with Knoblech to publish on brain organoids. And that's the thing that they found, that the epithelial cells in the choroid plexus are the main ones with the ACE receptors, and they die.
Okay, thanks
So,
for
I don't know about brain fog. So, but seizures you can explain, I think, when these epithelial cells die in the choroid plexus.
Okay, so Giselle is raising her hand. Yeah, Giselle, kindly unmute.
Seville, Kate, thanks. So Seville, we're in the area of pain research. You might know that we have an ongoing project with Toto Oliveira, where we culture mice dorsal root ganglion neuron. So my question has to do with systemic interactions between the peripheral nervous system and, of course, the central nervous system. So, is there a way to make use of iPSCs to model pain networks, pathways better? There's quite a bit of success in this area. We call it constellation pharmacology because we're able to culture the heterogeneous dorsal root ganglion neurons in the lab from mice. But then naturally, we'd like to do what you would consider a better in vivo model. And so right now, in the University of Utah with his collaborators, already involving many of my graduates who are doing their PhDs there with Toto, we actually look at the phases of pain generation, the initial, middle, and later phases, and then try to see what's happening with the cells, with a neuron, by doing transcriptome profiling over
Yeah,
time. So
yeah,
I'm just curious how we can, come up with maybe an easier model that's closer to in vivo, where, well, what's in
right,
vivo is the 3D structure of
right,
the tissues, right, or the organs.
right. That's right. Yeah, so I'm not familiar with, with the pain, or I don't know if people are generating those particular types of nerves from IPS. But as there have been quite a number of failures when you do research in, for example, mice. And people have seen, like if you treat the tumor in mice and it disappears, when you try it in humans, it doesn't work. So that's the reason why, I know what you're saying. If you can have a model that's really human based, then you probably and then also 3D. So I'm not sure about pain research. I'm not familiar with it. I can look into it, but probably I don't know. Have they looked into um, iPS-derived cells? Do you know?
It's not being done in
Not
our
being done.
group. So,
Maybe have they looked at whether other people are doing it?
so yeah, it's an area that should be explored. But
Yeah.
still, the other area is cancer.
Oh
So I'm
yeah.
sure this is a major area of application of induced pluripotent stem cells.
Yeah. Actually, this person in the Netherlands, he has done different types of organoids, intestinal organoids, kidney organoids, and stuff like that. He's
Oh.
one of the boy. His talks are like science fiction. The cells are moving, and he shows the organoids. I forgot his name. I can send you his name. It's in cancer.
Okay, thanks, Seville. So the
You're
next question comes from Ahmad Mazaheri. So he's asking, as you know, one of the known challenges of generating patient-derived iPSCs is the issue of differential propensity. Some IPCs, depending on the source, can differentiate better towards a tissue lineage than others. How do you overcome these issues?
Yeah, actually, this is one of the limitations of IPS. So even if you take it, even if your source is the same, from the same fibroblast, you reprogram, so you pick multiple clones, IPS clones. Though not every IPS clone that you pick is the same as the other one. So the best thing that to do is to use 3 different clones, or at least 2 clones. And actually that's what, from that same source, and measure, as I said, measure the, do karyotyping, make sure that there's no gross genetic defect in that has been carried by those iPSs. And just make sure that those iPSs are growing at about the same rate. You know, you don't want to pick one IPS clone that grows very slowly, the other one grows very fast. So from the same parental cell. So that's one of the limitations of IPS. But at this point, people are not talking about it too much anymore. I think Paul Liu at the NIH has done a very good investigation of these different clones. And actually, when you pick the right clones, there's not a lot of difference between those clones. That's what the take-home message was.
Okay, thanks, Seville. So I'll just take the last 3 questions.
Okay.
Because they're quite long. Okay, the next one comes from Dilberto Ferraren. So thank you for
Hi, Bert. How are you?
Okay. Okay, so
I did
thank
not
you very much.
I did not include your exact, what was your exact position there?
So,
Vice President for Planning lang po, ma'am.
Oh, lang, huh? Vice President
Vice
lang.
President lang. Assistant President.
Congratulations, Bert.
Okay, so his question is, you have expanded the tools in identifying the genes that result in SZD and BP disorder. Is it possible to simultaneously use 30 independent samples in different dish to grow iPSC, the samples, and compare their development behavior? Since the disorders are multifactorial and most probably are multigenic and thus is theoretically expected to have distinguishable morphological differences in the neuron.
Yes, actually, Bert knows that these are polygenic diseases. So that's the reason why you had to target the research from different angles. And as far as growing these cells in, some people grow them in 96 well plates. So you can studied 96 different clones at the same time. And also, this is one thing that I think that we will do in the future. People do a multiplexing. Like they combine, for example, 10 to 20 clones in one pool. So you do pooling and then compare the phenotypes, whatever types of assay you are doing. And for example, a pool of clones from patients versus a pool of clones from controls. And you can do several pools. That way you don't have to study them individually right at the start. So if there's any difference, then you can start, taking the individual clones from those pools. So it's similar to what NIH is doing as far as COVID testing is concerned. We get tested at NIH and your samples are placed in a pool, like probably a pool of 20 people. So if everyone in that pool is, has no infection, then you don't have any infection. But if there's anyone in that pool that has infection, then they, they take the samples from those other people individually. So, yeah, Bert, it's it's still a really major problem trying to determine the biology of these small effect variants. That's where we are.
Thank you for that, Seville. And then the next one comes from Al Serafica. So good talk, Seville. Hope you can retire here in the Philippines and set up a lab. What and how much does it take?
No, it's Francis. That's why we are training the next generation. Francis will be going back. Francis, say something. And he will be going back, and then, he can train his fellow students and the other students who will be interested in doing this project.
Okay.
Type of
And
project.
who was that?
Who is that?
Someone spoke. Okay, anyway. Okay. The next one comes from Lisa Verata. So nice talk. CDK RAP2 deficiency linked to microcephaly looked interesting. Do you think that in Zika infections it is possible for the virus to cause the protein deficiency in microcephaly?
In Zika, one of the first papers on Zika virus using iPS-derived neural stem cells or neural progenitor cells was from one of our collaborators, Guali Ming. She used to be at Hopkins, now she's at the University of Pennsylvania. What they found was that the virus attacks the neural progenitor cells and they die. So that's what they found. Does that answer your question, Lisa? Oh, maybe
Lisa?
she's.
Hold on. Yeah, Lisa,
Thank
yeah,
you.
there.
I was just throwing it out there, but
Oh,
yeah,
yeah.
that sounded very interesting
Thank
that they
you.
actually found a gene.
Oh, yeah.
Yeah, that
Not
could
gene,
cause
but yeah,
deficiency. Yeah.
yeah. Oh, for this microcephaly, that was not.
For causing
Yeah,
microcephaly. Yeah.
right,
Yes.
right.
Okay, thank you. I think that's all. Yeah, and
Oh,
it's
okay.
almost 9:30. Anyway,
Oh,
Seville, will it
really?
yeah. Seville, will it be okay with you to share with us your presentation, maybe even in just PDF form, so
Wait,
that I
what?
can your
Oh,
presentation, yeah, can
oh,
you
in PDF. I have it in PDF.
yeah, yeah, kindly
Okay,
send it to me
I'll
so that
send it to you.
I can share it with the participants. So do you have
Oh,
any final parting words for our audience?
oh, what is very important, I think, for us who are at this stage, especially me, I'm old, is to really train the next generation of researchers and scientists in the Philippines, because we need a critical mass. Why is it that Korea is very good at that? Why is it that other countries are much better than us in terms of, getting research going and stuff like that? So I think the more next young people we train, that's, I think, is one of the missions. Um, I think all of us share that mission and vision. Um, we can only we have over here we have limited way of um, contributing to that mission, but that to me I think is very important, because we cannot stay this way every year, every decade. We have to get more competitive out there in scientific research. It used to be, when the Chinese started to let students out
Mm-hmm.
of China, there was hardly any postdoc, NIH postdoc at China,
From
Chinese
China.
NIH, from China at NIH. Now you have like, I don't know, 30, 40% of people who are doing postdoc and at NIH are Chinese or students. It's just, you see this when you are in the parking garage. It's very noticeable. So I don't know, maybe we are not, are we science, are we interested in science, or are we, maybe it's just that we need to get more stimulation.
Yes. Agree. Lisa, did you have something to say?
Oh, and there's only a handful of Filipinos at NIH right now.
Also,
Yeah, that's right.
You
So
can
for those
count.
of you listening who are interested to do a postdoc, maybe you should get in touch with people.
Yeah, right. It's just, or even
Yeah.
in universities. So,
Yes. Okay.
well,
So that's the role of PAASE also. We try our best to link up individuals from the Philippines and of course those of you pioneering in the US. And we really hope that we can generate more researchers and scientists.
right.
Thank you so much, Seville, for that very interesting talk. But before we go, I just wanted to share with you our talk for next week. Okay, so this is our talk for next week. So we have Dr. Danilo Casimiro, who is the Vice
That's
President and
interesting.
Head of global research and external innovation at Sanofi Pasteur. So as you know, there are more and more, the research on vaccines is progressing very fast and, the companies are already rolling out some information on the efficiency of these vaccines. So if you'd like to learn more about that, so we'll have Dr. Casimiro next week. So please join us. To register, just go to bit.ly Casimiro. Okay.
Thank you. As I said earlier, Kay, I really wanted to tell everybody how much I admire your patience and your effort in getting these lectures going. And
Yeah, well, I want to thank
sustaining.
also all the speakers for saying yes, for saying yes, and yeah, for some of those that I need to go to whenever I don't have a speaker lined up, and they're all ready to give a talk the following week.
Yeah,
So
that's great.
PAASE has been really active recently. So last night there was an REC panel discussion in the, I think, economics section, and we're having one also this afternoon for those of you who want to join. It's at 3 p.m. Philippine Standard Time. So this is about understanding about what happened during the Typhoon Ulysses. So this is being spearheaded by CP David, and our speaker is Dr. Jane Perez. I think Dr. Jane is one of the winners of the ASEAN US Science Prize for Women way back. I'm not sure which year. And she worked with Dr. Joey Comiso, if I'm not mistaken. So we had her as speaker at the 2019 APAMS conference as well. So if you want to join, please register at bit.ly REC12_01. And again, we have our website, which Giselle is regularly updating at paase.org. And we're uploading our webinars at our YouTube channel, bit.ly/paase webinars. Okay, so with that, I'd like to thank everyone for joining us this morning, and I hope I'll see you again next week and in all other PAASE activities. Thank you so much, Seville. It was a very interesting talk.
Thank you so much, Kay, and good luck.
Yes, thank you.
Stay well, everybody.
Stay well. Thank
Hi,
you,
Bert.
Ma'am.
Bert,
Hi, good morning.
Ayun,
Or good evening po.
finally, I see you.
Okay.